Lac Operon, regulation at transcription initiation. The story of Lac Operon unfolded as Jacob and Monod began studying about beta galactosidase, a gene that could break down lactose into galactose and glucose. And as they began studying that, they were able to structure what is called as an operon and basically uh, show or refer to the fact that it is not the enzyme that can be mutated alone, but it can also be a gene that gives control or has control over the expression of the genes that can be mutated. And with that came up the idea that genes can be regulated. So let us look at the learning outcomes of this session. So the concept of operon and the switching on and off of the genes is well understood because of Lac Operon. And this was first studied, of course, by Francois Jacob and Jacques Monod. Now, the Lac Operon is regulated by both a repressor as well as an activator, but both of them respond to two different signals. So it would be interesting to look at how. The Lac Operon is a cluster of genes that is involved in the utilization of lactose, and all the genes are expressed in one go. So it is basically a polycistronic system, and therefore all enzymes required for the utilization of lactose are expressed at the same time. They are under the control of just a single promoter. Lac Operon is considered to be a regulatory system for negative control, for positive control, and also what is called as expression by induction. So let us look at all of these in this coming slides. So we look at the discovery of Lac Operon, of course, as mentioned just a minute back, that it is basically Francois Jacob and Jacques Monod who discovered the Lac Operon in E. coli as they were studying the beta galactosidase gene. So what they observed was primarily that the beta galactosidase production increased several folds when the media in which the E. coli were growing contained certain galactosides. So what was observed is that the galactosides induces the synthesis of the enzyme almost minutes after the addition of the galactosides. And also what was observed is that if these galactosides are removed from the media, then the synthesis of the enzyme halted. So therefore, the interpretation from this observation was that the gene that produces the beta galactosidase seems to be able to be turned on or turned off in response to the presence or absence of galactosides, and that becomes the environmental signals because they are present in the media. So this was something extremely important, and this is also very interesting. Now, let us look at what the function of the Lac Operon is as an overview. So the Lac Operon is obviously giving beta galactosidases, and beta galactosidases are enzymes are responsible for breaking down the beta galactoside linkage between the galactose and the glucose, giving rise to the monosaccharides galactose and glucose. Now, they were also responsible for uptake of lactose and thiogalactosides. So what does that mean? It means that the Lac Operon gives rise to a protein that is called as lactose permease, and that lactose permease lodges itself in the membrane of the E. coli, and it becomes a carrier for the lactose or galactosides, and through the lactose permease they enter the E. coli. The third gene that is associated, or the function that is associated with Lac Operon, is not as well understood as these two, but what has been observed is that this third function that is associated with the Lac Operon has to do with getting rid of toxic galactosides. So other than different galactosides, thiogalactosides can also enter the E. coli, and thiogalactosides are toxic. So to prevent the thiogalactoside from killing the E. coli, the Lac Operon has an enzyme that is responsible for converting the toxic thiogalactoside into a non-toxic form, thereby saving the E. coli from death when it is using lactose or when it is using galactosides. So therefore that is interesting. Now, another observation that Jacob and Monod did with the study of the Lac Operon is that inadvertently, whenever galactosides are added to the media of an E. coli, immediately beta galactosidase will be synthesized. So they are suggesting that this gene is getting induced. But they also observed that along with beta galactosidases, the levels of the lactose permease also increases. So that gene is also getting induced. And also the levels of uh, thiogalactoside transacetylase increases. Okay, so basically there is a connect between the three, and so their conclusion was that all the three genes are being controlled coordinated coordinately. That means together they are being regulated together. So that is something that they learned. Now, we will now look at the structural aspects of the Lac Operon and see how the functions are carried out of the Lac Operon. So let's consider that you have an area, this is only a part of the genome of the E. coli, which contains the Lac Operon. And we will first begin with the structural genes of the Lac Operon. So the first structural gene of Lac Operon is LAC-Z, the second is LAC-Y, and the third is LAC-A. All the three structural genes of the Lac Operon are under the control of just one promoter, which is called as the Lac promoter. So when RNA pol comes and binds to this, all the three are synthesized at one go. So it is basically polycistronic. Now, as per Jacob and Monod study, when an inducer is added, okay, you have the transcription happening and the RNA being formed. Now, using this RNA is the translation happening, and the translation leads to formation of beta galactosidase, lactose permease, and thiogalactoside transacetylase. So basically, LAC-Z produces beta galactosidase, LAC-Y is the gene for lactose permease, and LAC-A is the gene for thiogalactoside transacetylase. Now, the levels of these may vary depending on the efficacy of the translation process. So what is observed is you have generally the highest level of the three enzymes is beta galactosidase, followed by lactose permease, and followed by thiogalactoside transacetylase. Now, to understand these, therefore, and the functionality of the Lac Operon, mutagenesis was the mode of the study. So you mutate a gene and study the function. You will find if the function is lost, then what is the function of the gene that way. So they basically obtained a lot of mutants, and one of the mutants that they obtained, okay, was able to continuously synthesize all the three enzymes. Okay, now the catch over here is that all the three enzymes were synthesized at the same rate and in the same amount, and also in the absence of inducer, which means that the mutant without an inducer was able to synthesize all the three enzymes, but otherwise a normal E. coli needs an inducer to have these enzymes being synthesized. So what therefore the mutant was denoted as was I minus, which means an inducer is not required for the synthesis of the three enzymes. What was observed in the mutant is that the negative control was lost, because when you have the observation that the three proteins were formed only in the presence of inducer, it means that there is something that is keeping the Lac Operon switched off. So there is a negative control which is there, but in this mutant that negative control was lost. It means also that because the negative control was lost, it was continuously switched on, and also the amounts of the enzymes formed were equal, same. Plus, what is also observed was that the enzyme activity was completely normal. There was no change in the activity of these enzymes, which means that the mutation has not changed effectively anything with respect to the enzyme. But it meant that the mutation is at another point in the Lac Operon, and that other point in the Lac Operon seems to be affecting the synthesis of these enzymes. So therefore, by this and mapping the mutations, they found that the Lac Operon has another gene which is called as the LAC-I. And this LAC-I was under the control of its own promoter, PI. And this promoter was more or less constitutive, which means that the RNA pol can come and bind to it and initiate the synthesis of the RNA from LAC-I. So the product of the LAC-I is present in the E. coli at all times. It is not produced only when required. It is produced at all times. So that is something that was observed by this mutant study. Now, going further, therefore, what is understood is that the LAC-I is responsible for giving rise to a protein that is called as LAC repressor. Now, as a monomer, if you can see, this protein has two domains. This is one domain and this is the other domain. One of the domains is able to bind to the DNA and the other domain is able to bind to a ligand. So, effectively what has been observed is that the LAG repressors do not exist as monomers, but rather they exist as dimers or tetramers, and more so as tetramers. And the interaction of the the monomers is such that you will always have the DNA binding domain opposite to each other. So this is one DNA, the monomers over here have the DNA binding domain here, and the monomers at the other end forming the tetramer have the DNA binding domain over here. And the ligand binding domains were at the central portion. Now, the question arises is, if this is the protein that is controlling the expression of the structural genes, then the question is, how does it do so? So what was observed is that the LAG repressor is able to bind to a region overlapping the LAG promoter, and that region is what is called as the operator region, or it is also called as LAC-O. So this operator sequence is such that enables the LAC repressor to bind. So the LAC repressor is able to go and bind specifically to the LAC-O or the operator region. Now, when you have this LAC repressor bound to the operator region, obviously it is covering a large portion of the promoter region as well. And since it is covering a large portion of the LAC promoter, even if an RNA pol tries to come and bind to the LAC promoter region, it is unable to do so because the LAC repressor acts as a barrier. In such a scenario, therefore, you will not have transcription being initiated at all because the RNA pol is not even able to make a interaction with the LAC promoter well. If it does not make an interaction with the LAC promoter well, it cannot change its conformation from closed to open, and therefore initiation will not begin. Now, the next question that is asked therefore is it really only the operator sequence to which the LAC repressor binds, or can it bind to any other place in the Lac Operon? So, to ensure that what was carried out is mutations at the operator region, and then they check the binding of the LAC repressor to the operator region. So the observations were that if there are mutations, even a single nucleotide mutation at the operator region, then the affinity with which the LAC repressor binds to the operator region diminishes. And so that was confirmed that the LAC repressor has an affinity to bind only to the operator region. Now, to look at the operator region more closely, what was observed is that the operator sequence is basically a 21 base pair, two-fold symmetry structure. What do we mean by two-fold symmetry? If you have a line of symmetry over here, then these two are kind of mirror images, but inverted kind of. Now, 10 base pairs. So this is one 10 base pair comprising a half site and the other 10 base pair comprising another half site. If you read this AATTGTGAGC, you will see that when you read it from here, you will read it almost the same AATTGTTATC. Here there's a difference of only one nucleotide, that can happen. But otherwise, more or less, they read the same. So that is why they are called as twofold symmetry. This is strategic. Why is it strategic? Because every monomer can now bind to one monomer can bind to one half site. So in a tetramer, a dimer is able to bind to one half site each. So you have the DNA binding domains of the Lac repressor specifically interacting with the half sites, not anywhere else. It interacts with the half sites. So the question was, what is there in the DNA binding domain that enables it to only bind to the half sites? So on further study, it was observed that these DNA binding domains have a very typical DNA binding motif called the helix-turn helix motif. And of the two helices present in the helix-turn helix, one of the helix is a recognition helix, which has a set of amino acids that can specifically interact with the nucleotide sequence of the half site. And it is able to interact with the sequence in the operator region through the major grooves. The other helix of the helix-turn helix is only needed to basically hold on to the DNA. So this is how a helix-turn helix over here interacts with one half site, and a helix-turn helix here interacts with the other half site. So therefore, the interaction is quite stable. Now, what question next comes is, why a tetramer when you only need a dimer to bind to the operator region? So let us look at this aspect as well. So what has been observed is that in E. coli, other than the Lac O, which is the real operator sequence and that lies exactly in the Lac promoter region, there are other pseudo operator regions also in the E. coli DNA. And the pseudo operator regions are either upstream or downstream. So if you go further over here, you will find another pseudo operator region over here. Now, when I say pseudo operator region, it means that the sequence is very similar to the Lac O, but it is not the real operator region. But because the sequence is very similar, there is homology. The Lac repressor is able to bind to the pseudo operator region. So what advantage does that have? So here, what you observe is if this is present upstream, so this, if you straighten the DNA, you will find that the pseudo operator is present here. Just because the DNA has bent, you find that they are juxtapositioned opposite to each other. Now, when they are juxtapositioned opposite to each other, what you find is that the entire tetramer can bind on one end to the real operator and the other end to the pseudo operator. So, how is this conformation now helpful? Well, because this Lac repressor as a tetramer can bind to the pseudo operator as well, it is increasing the level of repression. Its negative control is better or more efficient as a tetramer. So why so? When you have the DNA looping over here, there is a steric hindrance. So this steric hindrance, so if the RNA pol wants to come and bind to the promoter region over here, it has now greater steric hindrance. It has the DNA looped over the Lac promoter region, so the RNA pol finds it even more difficult to come and bind to the promoter. So the accessibility of RNA promoter to bind to the Lac promoter region decreases further on the Lac repressor binding to the pseudo operator regions as well. So interesting therefore is that the helix-turn helix is present on the extremes of the tetramer, and at the center over here, as mentioned earlier, these domains of the monomers are able to bind to inducers, or what is called as galactosides or lactose, more consistently allolactose, which has structure similar to galactosides. So these can bind to these sites in the tetramer. On binding to these sites, what is observed is the moment the galactosides bind to these sites, you will find a conformational change in all the monomers. So this conformational change is very important. Why? Because as you can see, the DNA binding domain conformation changes, and in this conformation, its affinity to bind to the DNA decreases. So with the inducers present on the Lac repressor now has minimum affinity for the operator regions, whether it is pseudo operator or whether it is the Lac operator, and the repressor will move away from the operator region. So affinity for half sides decreases. So let us look at therefore this scenario that we have understood is that if there are no inducers, that is the media has no galactosides present, then the Lac repressor gives rise to the repressor, the Lac repressor formed by the Lac I can go and bind to the operator region. When it goes and binds to the operator region, it acts as a barrier, and therefore the RNA pol is not able to position itself properly on the Lac promoter, and hence you will not have transcription happening, not have transcription initiating. Now, let us look at the other scenario where you have the inducers in the media. So when the inducers are present in the media, obviously the inducers are going to bind to the site where you have the ligand binding site in the Lac repressor monomers, and as soon as the inducers bind to this, you have a conformational change in each of the monomers, okay, by lieu of which its affinity to bind to the operator region decreases and it basically leaves the operator region. So therefore it is open for now the RNA pol to come and bind to the promoter region and initiate transcription. It is open, okay. This concept of a ligand binding to a site on the protein and changing its conformation and thereby its function is what is termed as allostery. So this is extremely important as a phenomenon in control of regulation of gene expression. So binding of inducer to all the monomers changes the conformation of all, and that is not conducive to bind to the operator region. This is important. So in this case where there are no inducers, the Lac Operon is switched off. In the case where there is inducers, the Lac Operon is switched on. Now let us look at one another aspect that is there with the Lac Operon, and that is with relation to the Lac promoter itself. Like many promoters, Lac promoter has a -35 sequence and it has a -10 sequence. But what has been observed is that the -35 is deviating from the consensus sequence, and therefore when the RNA pol comes and binds to the promoter region, the sigma 4 subdomain of the sigma subunit is not able to interact that well with the -35. So the interaction that happens of the RNA pol with the Lac promoter is a weak interaction. So the RNA pol can form maybe a closed complex, but it has difficulty to go into open complex because its interaction with the promoter is weak. Now, so the question that had come up is then how is it that the RNA polymerase initiates the transcription? So what was observed is that within the Lac operon, just upstream of the Lac promoter is present a sequence that is called as the cap binding sequence or the CRP binding sequence site. What is cap? Catabolite activator protein, or CRP, that is cyclic AMP receptor protein. It is one and the same. Some call it cap, some call it CRP. Now, this binding site is specific for another molecule called cap protein, and that cap protein is basically an activator. So it can come and bind to the cap binding site, and on binding to the cap binding site, it has a conformation that enables it to recruit the RNA pol, and on recruiting the RNA pol, because now the complex is activator, RNA pol and the Lac promoter, it forms a stable interaction. So the activator is responsible for enabling the RNA pol to form a stable interaction, and when it forms a stable interaction, a closed complex due to the phenomenon of cooperative binding, okay, so this activator recruits the RNA pol is what is cooperative binding, and that forms an open complex, so transcription initiation can begin. Here it is an important thing to note, even if the repressor is absent, okay, say in both these cases we did not look at the repressor present at the operator region at all. So if the repressor is not present in the operator region, the RNA pol is able to come and bind. But see, look at the fact that it cannot initiate replication on its own unless and unless the cap comes and binds. So this is what is called as positive control. That is, the cap initiates initiation and therefore it is a positive control, while the Lac repressor binds to the operator region and inhibits transcription initiation, and therefore that is called as negative control. Also, just before this, we looked at the fact that the galactosides as ligands can come and bind to the Lac repressor and change its conformation. That is what is called as expression by induction, because in that form the Lac repressor was not able to bind. So the galactoside binding to the Lac repressor induces the initiation of the Lac Operon. So that is expression by induction. Now, just understanding this concept more about cap being able to carry out cooperative binding. So the cap has a binding domain for the cap site. And when studied further, it was observed that like Lac repressor, even the cap has helix turn helix as a DNA binding motif. And it is able to, when binding to the cap site, also bend the DNA such that the DNA winds around the cap. So the interaction becomes quite strong. And because its interaction is quite strong with the DNA, it is able to recruit and position the RNA polymerase right at the promoter region, which is required for the stable interaction and opening of the RNA, that means the DNA, unwinding of the DNA, formation of the open complex. Now, understandably, the question that was asked was, how is the cap recruiting the RNA polymerase? So what was observed is that cap also has a domain that can bind specifically to the RNA polymerase, and the binding is specifically with the alpha CTD tail, the C-terminal domain of the alpha complex of the RNA polymerase. So because the alpha CTD can bind to cap, the cap can hold on to the RNA polymerase at the promoter region. Looking at the structure more closely, you can see that the cap is also a dimer and it has very specific domains. So this domain is what is the D helix, that is the DNA binding helix, and you have typically the helix turn helix structure. This becomes a recognition helix, and this is basically one that holds on to the DNA. So the helix turn helix can bind to the DNA only and only when cyclic AMP is bound. So you can see that there are certain points in the protein where the cyclic AMP can bind. So it's only when the cyclic AMP binds to the two monomers that the helix turn helix is exposed to bind to the DNA. Suppose you remove the cyclic AMP from these regions, then there is a change in the domain at the D helix, and the helix turn helix is positioned internal. So it is present inside and not exposed outside. So this cap cannot bind to the cap binding site any longer. So you can very clearly see over here that the binding of cyclic AMP to the two monomers changes the conformation such that before binding it is in an inactive form, and after binding the cap is in an active form, and it can bind to the DNA at the cap binding site. Also, with the binding of the cyclic AMP, what is observed is the C helix. This C helix is the helix that binds to the C-terminal domain of the RNA polymerase. That too is defined well because of the conformational change, and it is able to bind to the alpha CTD tail. So at one end, the cap binds to the DNA, and at the other end, it binds to the RNA polymerase, and thereby it carries out both cooperative binding as well as it shows what is called as allostery. Now let us look at certain aspects related to the Lac Operon. So when cyclic AMP is high, it can bind. So there are lots of cyclic AMP available in the E. coli, and they can bind to cap. And when they bind to cap, cap is active. But because cyclic AMP has no association with the Lac repressor can also be active. So here what you can see is that the negative control is active or is present, but you also have the positive control present. But like we had seen in basic principles of regulation of gene expression, if the Lac repressor is present, then it is generally that you have the Lac operon switched off. Now, this happens when the sugar content is low and the ATP is decreasing. So, then you have the cyclic AMP high. The other scenario is cyclic AMP is low. If cyclic AMP is low, is CAP is inactive, but Lac R is active. When does this happen? This happens when sugar content in the media is high, and that is when the ATP within the E. coli will be high because the sugar will be utilized to form ATP. So effectively what we understand is that the ATP-AMP ratio is critical. When the ATP rises, AMP decreases. When AMP rises, ATP decreases. And that ratio is what is responsible for 1/2 of control of the regulation of gene expression in Lac Operon. And of course, the third scenario is when you have an inducer present in high content in the media, then you have the Lac R inactive. So the negative control is abolished. So there can be, therefore, from all of these understandings, two scenarios. One, you have a high level of cyclic AMP. That is because the sugar content in the medium is low. Okay. You mean to say, therefore, that there is a particular sugar content that is low in the media, but lactose is present. And if lactose is present, then LAC-R becomes inactive and CAP becomes active. So positive control is on, negative control is off. In such a scenario, definitely you will have the Lac Operon on. Other instance can be the cyclic AMP is low, the CAP is inactive, lactose is absent and LAC-R is active. In such a scenario, Lac Operon will be off. Now the question to be asked is, when will you have such situations? And also, what is clear is that there are two signals. One is that of cyclic AMP and the other is that of the inducer. So, to understand this, therefore, we go to an experiment that was carried out by Jacob and Monod itself. And in that experiment, what they had done is they had grown the E. coli in media containing glucose and lactose. And of course, the study has been carried out by several other scientists as well. And what was observed is that in presence of glucose and lactose, the E. coli growth curve was very different from when E. coli was grown individually in media containing glucose or containing lactose. Now, the general perception would be when there are two carbon sources available, then E. coli will use both the carbon sources together. But the observation that they obtained from the results was very different and also interesting. So the growth curve that they obtained was that you have a sudden growth in the E. coli, then you have a plateauing of the growth, and then after a lag period, you again have the growth of the E. coli growing. So you can see you have two exponential growth phases in this growth curve of E. coli. So the interpretation of this was that E. coli would preferentially use glucose first. So therefore, the first exponential growth curve that is obtained is because of glucose utilization. And then once glucose was completely utilized, after a certain plateauing of the growth, and as the E. coli began to take up lactose for its carbon source, you would find that then you would have a growth again, exponential growth curve again, and that was an indication that lactose is being utilized. So this is what is referred to as the dioxic growth curve. Okay, and interestingly, therefore, what comes out from this is that while glucose is being utilized, lactose is not being utilized, and lactose is being utilized only when glucose is not there. So effectively one can say that the Lac Operon is active only and only when glucose has been completely utilized. If the media has glucose, then it is not going to be switched on. But if the media has completely been depleted of glucose and only lactose is available, then the Lac Operon gets switched on. So the understanding is that when glucose is being utilized, ATP is high because glucose is broken down into carbon dioxide and water, and in the process a lot of ATP is being produced. So the energy currency in the E. coli is high. If the energy currency in E. coli is high, obviously the CAP is going to be inactive because AMP is going to be low. And if CAP is inactive, it cannot recruit the RNA polymerase much. But what has been observed is that the RNA polymerase does or sometimes come and interact with the Lac Operon and synthesize. So there can be a basal level of transcription present despite there being the presence of lac repressor. So effectively, positive control is absent, that is, cap cannot act, and the lac repressor is acting, so negative control is present, and hence lac operon will be switched off when the media has glucose. Look at this scenario. During the lag phase, as the E. coli begins to take up lactose, because there is a basal level of transcription, there will be some lactose permease on the surface of the E. coli and it will take up lactose. So when a certain amount of lactose is accumulated within the E. coli, then the lactose will bind to and in fact it is converted to a form called allolactose, and that allolactose will bind to the lac repressor, and on binding to the lac repressor it becomes inactive. So here what is observed is negative control is absent, and plus because all glucose has been utilized and also the E. coli is trying to survive, it is utilizing all the ATP, and so in the ATP AMP ratio cyclic AMP becomes high. When cyclic AMP becomes high, cap becomes active, and then therefore cap can recruit the RNA polymerase and carry out transcription, which means that during this phase of the dioxic growth curve, lac operon is switched on. And effectively, the signals that the lac operon has responded to is as external signals the glucose and the lactose, and as internal signals it is the cyclic AMP and the allolactose, and this allolactose is formed from the lactose itself. So to get a final overview of the lac operon all over again, okay, let us look at the scenario where glucose is present as well as lactose is present. What does it mean? That means E. coli is growing in a media containing both glucose and lactose. In such a scenario, what is going to happen is lac I is going to produce the repressor. The repressor is going to go and bind to the operator region because you have very negligible amounts of lactose being present inside. Since glucose is being utilized by the E. coli, the cap is there, but because glucose is being utilized, ATP is high and AMP is low, so it will not bind to cap. So therefore, effectively, although RNA pol tries to come and bind the lac promoter, it is not being recruited by the cap because cap is inactive, and it is not being allowed by the lac repressor to bind to the lac promoter because it is bound to the operator region. So the negative control is present, the positive control is absent, and an effect of that is that the lac operon is switched off. And like I mentioned, it is possible that you have basal levels of transcription happening because the lac promoter is considered to be a leaky promoter. Well, if you look at an analogy, say for example, you have a tap. So a tap can be switched off, a tap can be put off. Okay, you can turn the tap to have the water flow completely stopped. Now, suppose there's a problem with the washer of the tap. Even if you close the tap, you can find that water can, go out as drops. So that is considered to be a leaky tap. Similarly, is the case with lac promoter. So sometimes what can happen is that the lac repressor is not bound to the operator region, and the RNA pol is able to make a weak interaction, and while making a weak interaction it may go from closed to open and then suddenly synthesize a few mRNA, and therefore few beta galactosidase, few lactose permease, and few thiogalactoside acetyltransferase. But then repressor again comes and binds and it gets switched off. You see, so it is leaky. So effectively what we are trying to say is that some level, very negligible, but some level of beta galactosidase, lactose permease, and thiogalactoside acetyltransferase is present in the E. coli even when the lac operon is switched off. So a little amount of lactose can be taken up. Now let us consider that all glucose has been used and what is present is only lactose. So if glucose is completely absent, what is going to happen is lactose is going to slowly gradually accumulate in the E. coli because there is presence of lactose permease on the membrane of the E. coli. Now, when a certain level of lactose builds up, in the E. coli, only then it will go and bind to, because you see, you need already, if you effectively look at one lac repressor, you need four lactose molecules to make it inactive. If you have several lac repressors, you need that many number of lactose to bind and shut off or change the conformation of the lac repressor, so it does not go and bind to the operator region. So therefore, there is a lag period in the dioxic curve, you see. So in this scenario, what happens is, since the glucose is absent, ATP is being continuously used up by the E. coli for its various survival metabolism. AMP increases in the E. coli, so AMP can come and bind to cyclic AMP active receptor protein, and this then comes and binds to the cap binding site. When it binds to the cap binding site, its conformation changes such that it is able to recruit the RNA pol. And on the other hand, at the same time, because of the presence of the basal levels of beta galactosidase, the lactose that has been taken up by lactose permease in the membrane of the E. coli is being converted to allolactose by beta galactosidase itself, and allolactose can go and bind to the lac repressor with greater affinity. So when they bind to the ligand binding sites of the lac repressor, obviously the conformation changes and the lac repressor falls off. And if you have many more lac repressors, all of them will be bound to by allolactose, and so all lac repressors are inactivated and they don't go and bind to the operator. So from both ends, the RNA pol is now can get recruited, and secondly, there is no barrier. So the RNA pol gets into a stable interaction with the lac promoter. It gets converted from a closed complex to an open complex, and the Lac Operon gets switched on. So this is how it is working with two signals. The cap, that is responding to levels of cyclic AMP, which in turn is responsive to glucose being present or not. The lag repressor, which is responding to the presence of the inducer, and that in turn depends on whether lactose is present or not in the media. So you can see how both the regulators are responding to two different signals. So let us make the conclusions. Lac operon is a cluster of genes dedicated to utilization of lactose. When lactose is present, only and only then the lac operon becomes on. The lac operon is regulated by an activator, which is cap protein, and by a repressor, which is lac repressor. So the cap is active when it is bound to cyclic AMP, and the lac repressor is active when it is not bound to the ligand. So you can see allostery over here. The lac repressor keeps the lac operon switched off by binding to an operator region that overlaps the lac promoter, and hence it doesn't allow the RNA pol to come and bind to the promoter region and initiate transcription. However, when inducer binds to the repressor cannot bind to the operator region, and the whole thing gets reversed. Cap cyclic AMP recruits RNA polymerase to the lac promoter and initiates transcription when lac repressor is not bound to the operator sequence. Thus, Lac Operon, discovered by Jacob and Monod, is a paradigm for gene regulation with two signals, one positive and one negative, controlling the gene regulation. And of course, induction is a phenomenon of control of regulation over here because of the inducers like galactosides. Thank you.